Welcome back, students. So let me continue with the electron transport chain. As I was talking, electron transport chain, electrons are transferred through a series of redox pair which are arranged in such a manner that the redox potential of each redox pair as you go in the electron transport chain is more than that of the previous one. As the redox potential is increasing, that is how this ETC is arranged. That is why electrons can keep moving. Now I showed to you how from NADH it has gone to complex 1 which contains FMN and iron sulfur. From this it has gone to coenzyme Q. Complex 2 also has FAD, iron sulfur, and then to CoQH2. It has formed CoQH2. Now it has given it to complex 3. Complex 3 also has cytochrome B and iron sulfur and cytochrome C1. MCQ point of view, let us remember B and C1 are first B, then C1, and now cytochrome C. Let us talk about cytochrome C. Remember, in this CoQ is a ubiquitous lipophilic mobile electron carrier. Cytochrome 3 is the enzyme, CoQ, cytochrome C reductase, and it also has an iron sulfur center, where iron is again a redox pair, ferric to ferrous, ferrous to ferric is taking place. Now cytochrome C, what are its characteristic features? Cytochrome C is loosely bound to the inner mitochondrial membrane. Now it is also mobile and it can be easily extracted. It is a hemoprotein just like all the cytochromes. It transfers electrons from complex 3 to complex 4. So cytochrome C takes up from the complex 3 and from complex 3 it gives it to complex 4. So I'll just write it here, complex 4. Cytochrome C is now going to give it to complex 4, the electrons. So what is happening in complex 4? What is this complex 4? Complex 4 is the one which is going to react directly with oxygen. It is called as cytochrome oxidase. Remember, this is cytochrome reductase, this is cytochrome oxidase. Here it has reduced cytochrome C, so it is cytochrome C reductase. Here it is going to react with oxygen, so it is cytochrome oxidase. Cytochrome oxidase. So it contains the cytochromes A and A3. AA3. It transfers electrons from cytochrome C to molecular oxygen. Now, whole oxygen molecules must accept 4 electrons to be reduced to 2H2O. So, that is the way it can be balanced. To tell it simply, we usually write it as half O2 to form H2O. Okay, it has cytochromes A and A3 and iron sulfur, and plus it is a copper containing. I'll talk more about complex 4. It gives the electrons. Final electron acceptor is oxygen to form water. So we have seen how electrons are transported from NADH to oxygen and finally water is formed. Okay, so a whole oxygen molecule must accept 4 electrons to be reduced to get 2 molecules of water. That will be the balance between them. So let us look at it, how it is happening. It is taking up cytochrome C is giving it to cytochrome AA3, which is getting converted and finally to half O2 to form water. So let us know more about this cytochrome oxidase. It is reacting with oxygen. It is a special thing. It is the only enzyme within the electron transport chain which contains copper. It is the only enzyme in which iron directly reacts with oxygen. Now each oxygen accepts 2 electrons and accepts 2 protons from the medium and a molecule of water is formed. This water that is formed is called as the metabolic water, and the metabolic water is around 300 ml per day. And this is the only irreversible reaction in this chain. Cytochrome oxidase has got very high affinity for oxygen. So all the ETC will function at maximum rate until the tissue becomes depleted of oxygen. Oxygen levels become the rate limiting factor. If oxygen is low, only then cytochrome the electron transport chain may not function. So long as there is enough oxygen, the ETC will function at the maximum rate. Okay, so now let us see further. Right now we saw how electrons are transported across a different redox pair. Now I told about all this. Where is the ATP synthesis in all this? We keep talking of electron transport chain, electron transport chain. Why the body has to do such a long procedure? Keep transferring electrons from here to there, here to there, here to there, and finally give it to oxygen. And what if no ATP is formed? The purpose of this was to form ATP. And where is ATP formed in all this? It is for this, different scientists worked out on a lot of problems, and then finally one of the accepted theories that has been accepted now is the chemiosmotic theory given by Peter Mitchell. So let us look at this coupling between the electron transport and ATP synthesis, how these are going to be linked, and this is given by the chemiosmotic theory by Peter Mitchell. So how electron transport is linked with ATP synthesis. So let us look at the chemiosmotic theory. Now before we go further, let me just tell you how it is happening. Now the free energy during this transport, we keep talking of free energy. That's why I said bioenergetics I talked about, wherein I talked about free energy, the Gibbs free energy. So during this process of transport of electrons across the electron transport chain, what is happening? A lot of free energy changes are taking place. So this free energy is harnessed for the formation of ATP. This free energy is coupled to the formation of ATP. So there are what are called as coupling sites. Let us know that coupling site for ATP synthesis includes complex 3, complex 1, complex 3 and complex 4. Complex 1, 3 and 4 are the coupling site. Complex 2 is not a coupling site. Okay, with this background, let us see complex 3 and complex 4. So electrons from NADH pass through all the 3 sites and thus yield 3 ATPs, whereas from FADH to bypass complex 1 thus yield only 2 ATPs. From C2 complex 2 from FADH to go only through complex 3 and complex 4, hence 2 ATPs. Whereas from NADH it goes through all the 3 coupling sites, hence we get 3 ATPs. So let us look at it. Before we go further, how chemiosmotic theory, let us look at one more complex that is known as complex 5, which is the site for ATP synthesis. This is also called as FOF1 ATPase. Let me show you this, how it looks like. A bit about FOF1 ATPase, the site for ATP synthesis. Remember, I told you the inner mitochondrial membrane is folded into cristae. Now, in these cristae, after the electron transport chain, there are 4 complexes. We have one more complex, and this is somewhat shown like this. Okay, with a knob and a head, pinhead appearance with all this. These cristae, complex 5 is located in the inner mitochondrial membrane and it is shown like this. This is the inner mitochondrial membrane. It goes on, as you can see. So these are arranged in this cristae. This is called as FOF1 ATPase or ATP synthase also. It is made up of 2 subunits, FO and F1. O is for oligomycin. Please note that it is not F0, F1. It is FO and F1. Why O? O is for oligomycin, which is an inhibitor of this subunit. It has 2 subunits, FO, F1. Now FO is embedded in the inner mitochondrial membrane and it forms a channel for the passage of proteins. So as I said, it is here. So if you look at FO, it is, if this is the inner mitochondrial membrane, the FO is embedded. It is a part of this protein is a part of the inner mitochondrial membrane, and it has a pore in it through which the and it goes ahead, and this is the second subunit, F1 subunit. So FO is a part of the inner mitochondrial membrane, IMM. This is embedded, this is FO, inhibited by oligomycin, this is F1. Now let us look at it. It forms a channel for the passage of proteins. F1 is tightly bound to FO, but it protrudes into the matrix and it contains the catalytic site for ATP synthesis. This is actually complex 5 is considered as a molecular motor. It is more like a motor. It is having a complex structure. Everything, the protein structure is very complicated, but it is acting like a molecular motor where some things are turning and twisting, which helps in the synthesis of ATP. So let us look at the mechanism, the chemiosmotic theory. I kept talking of how electron transports, the free energy is harnessed. If I just say that the free energy is harnessed, it doesn't make sense. How is it harnessed? At the coupling sites, I told you there is what is called as a coupling sites also. Now let us look at how this is coupled. As I said, the inner mitochondrial membrane where the complexes are situated. So I'll just show it here: complex 1, complex 3 and complex 4. That is the coupling site 1, 3 and 4. Complex 2 is also there. Let me just not talk about it. Now as the electrons are passing to, again you have got oxygen here, which is forming water. So as the electrons go from 1 to 3 to 4 to finally to oxygen, what is happening at the coupling site? There is pumping of what? Of protons. Okay, the protons are pumped at these coupling sites from what? From the matrix to the intermembrane space. Remember, from the matrix, this is the mitochondria, outer membrane is there. Between the inner membrane and the outer membrane, there is what is called as the intermembrane space. So as the electrons go like this, it is coupled with the pumping of protons. So what will happen here? What will happen if protons are pumped like this? What will happen is this protons accumulate at the intermembrane space. So what? If protons accumulate like this, what is happening? What is the difference between here and here? If you look at it, one, the protons have got two things happening to them, have got two properties. One, they have a acidic property, so they can decrease the pH over here and there will be increased pH here. At the same time, what else it can do? It is having a charge also, so there will be a voltage difference. So that is what is the term. It is having a chemical potential, at the same time it is having a voltage potential, a electrical potential, and a voltage potential is also will be created between across the membrane. A potential is electrochemical potential is created across this membrane. So let us talk about this, the chemiosmotic theory given by Peter Mitchell. So the transport is coupled with the pumping of protons from the matrix to the intermembrane space at the coupling sites 1, 3 and 4. Remember, the inner mitochondrial membrane is impermeable to protons, so it results in higher concentration of protons in the intermembrane space. So it results in the generation of electrochemical potential gradient across the inner membrane. So it is like a big dam, huge dam is there, water is collected, but instead of water, what is there? It is the protons. So there is a wall over there. The protons all want to go across. They have that much potential energy between in them. That much potential energy is present in these protons and they all want to go. Suppose there is a dam and there is water. If there is a hole, what will happen? They will gush through that, all that water will come out through that hole. Now here is something happening there. There is a complex 5 and these protons want to go through that complex 5. That complex 5 has a pore in it, has a hole in it, has a pore in it. So this potential drives this proton to enter back here. This proton wants to come inside here. Okay, so as it is going, it will gush. It is like a huge dam which is having a hole. Then all that water will come out with a force. This force is ready enough to generate ATP. So let us look at it. This proton wants to come inside. So this electrochemical potential gradient is there. So the outer side of this becomes relatively more positive than the inner side. This is called as a voltage gradient. At the same time, because of the another characteristics, it becomes more acidic, what we call as the pH gradient. This whole thing is called as this electrochemical potential is also called as the proton motive force. Protons movement, proton motive force. So due to this proton motive force, the protons, ions flow back into the matrix down its electrochemical gradient through the pore of F4 and of complex 5. So as the protons move into the matrix, there is release of free energy, and this free energy is utilized by this enzyme. It is an enzyme, so it can utilize for the synthesis phosphorylation of what? ADP plus Pi to form what we wanted, ATP. So ATP is formed. We have the formation of ATP, what we wanted. So here there is coupling of electron transport across the ETC with a formation of an electrochemical gradient. The protons move and there is synthesis of ATP. Coupling between ETC and oxidative phosphorylation. So this free energy and this formation of ATP is catalyzed by the F1 subunit. So F1 ATP is actually consists of three parts: a rotor, a knob and a rod. I'm not going so much into the details. It is a molecular motor, and the current caused by the H plus causes the rotor and the rod to rotate. This rotation activates the catalytic sites in the knob where ADP and Pi are combined to make ATP. There is what is called as the bind change mechanism. Let us not go so much into the details. Just remember that F1 catalyzes the phosphorylation of ADP to form ATP. During this, the proton motive force, the free energy that is present, is used for the synthesis of ATP. So let us look at the transport. ATP is formed here. It has to go outside where it is required. Do we need a transporter? Yes, we have what is called as the ATP-ADP translocase. This is transported out by the help of adenine nucleotide translocase. This is a transmembrane protein which is specific for ATP and ADP. So ATP has been synthesized within the cell. So ATP has been synthesized with the coupling of electron transport chain and oxidative phosphorylation. Now let us look at how this whole thing is regulated. Regulation of oxidative phosphorylation. We call this as the respiratory control. Please look at this side. Normally electron transport is tightly coupled with oxidative phosphorylation. So anything which regulates electron transport also regulates oxidative phosphorylation. So in the presence of excess substrate like NADH or FADH and oxygen, respiration continues until all ADP is converted to ATP. So ADP is the rate limiting factor, and it is the ADP levels which control the rate of oxidative phosphorylation. Are there inhibitors? Yes, there are inhibitors of this. Inhibitors of the ETC proper, inhibitors of the oxidative phosphorylation or F1 ATPase, and uncouplers. Again, MCQ point of view, let us just look at it, how each of these are acting, where they are acting. MCQ point of view, I have given the electron transport chain here. Inhibitors of complex 1, amobarbital, pyrisidine A, rotenone. Inhibitors of complex 2, dimercaprol, antimycin A. MCQ, that's all that you need to know. Complex 4, cyanide, carbon monoxide and hydrogen sulfide. and inhibitors of ATP synthase complex 5. They block phosphorylation. We already know FO. O stands for oligomycin. Then you have got DCCD, dicyclohexyl carbodiimide. Just again MCQ point of view, that's all you need to know. Tetracycloside inhibits adenine nucleotide translocase. Now I come to an important thing known as uncouplers. Uncoupler, where was coupling happened, only then you can uncouple. So coupling has happened between the electron transport chain and the oxidative phosphorylation. So anything which can dissipate this proton motive force can act as an uncoupler. What can decrease the proton motive force? Anything which can allow the proton to come back into the matrix, or anything which can change the potential difference between across the membrane. We call this as uncouplers. They prevent the formation of electrochemical gradient. They dissipate or prevent the formation of the proton motive force. They allow electron transport to take place. They make the membrane abnormally permeable to protons. So you have, instead of one hole through which they have come, there are multiple holes. There is no dam at all. The dam is riddled with holes. All the protons, all the water keep on coming across. So there is no proton motive force. If there was a dam with a single hole, only then this FO, F1, ATPase could have worked. So here there is nothing, everything is moving across the inner membrane, is abnormally permeable. So these are all lipophilic compounds capable of binding protons, and the energy produced by the transport of electrons is released as heat and not used for ATP synthesis. Now these include just a name of the drugs and the different uncouplers. MCQ point of view, very important: 2,4-dinitrophenol dicumarol salicylate, bilirubin, thyroxine, thermogenin, and ionophores like valinomycin and gramicidin. Let me just talk you about thermogenin. Is mainly present in the mitochondria of brown adipose tissue. It is also called as the uncoupling protein. Again, it is dissipated as heat and help to maintain body temperature, what we call non-shivering thermogenesis. This is abundant in newborn and hibernating animals and maybe in thin people. That is why thin people may never go fat. Now ionophores, they include ion carrier molecules other than protons like valinomycin, which allows passage of potassium ions across a membrane. Gramicidin is an ionophore for sodium and potassium. Again, no positive charges. It will lead to dissipation of the proton motive force. So they abolish the membrane potential. So with this we come to the end of the electron transport chain and the different mechanisms by which ATP is synthesized, substrate level, and the coupling of the ETC with the oxidative phosphorylation. Thank you.